since the pandemic when I went back to my city.
Tell me, how have you been?
I'm a bit sad. I came back to Monterrey waiting to see the mountains and all the mega biodiversity, but look at it, full of contamination.
You're right. Bad air quality turns out to be really harmful for the environment and also has a negative effect on people's health.
In fact, last weekend I climbed Chipinque Hill and it looked really frightening. I'm terrified just thinking that we breathe air in these conditions.
Speaking of which, there's a gigantic smoke cloud right here in front of us.
Hey guys! I couldn't help it but to hear your discussion. What are you guys worrying about?
Hey there. Well, we are concerned by the state of air on the city of Monterrey.
Oh, then I have some good news for you. Come with me to my lab to see what the FCV UANL agent team are up to. Come with me! Hey guys, look, this is Mauricio, and he will be explaining our project to you. Yeah, sure, come with me. Hi guys!
You see, what the team is trying to accomplish is design of a biofilter system aiming to reduce the concentrations of air pollutants.
That's really interesting. How are they gonna do it?
That's a really good question. We are making the biofilter with bacteria using synthetic biology principles to decompose carbon and nitrogen gases.
That sounds great, and the fact that you're working with Monterrey's air is a solid base for future work in many other regions worldwide.
Of course! Oh, wait, look at the time. I have to feed my cats. See ya!
This team has really good ideas.
Yeah, I hope they do well in the future. But for now, let's go home.
Oh no!
I think we're locked!
No!
Thank you. So now please raise your hands. We'll start with the judges if you have any questions you would like to ask, and then with the teams, so they can start answering questions. Yes, Juan Pablo?
Yes, I would like to know how to prevent that other contaminants from the air affect the viability of the microorganisms that you use for the filter.
I'm sorry, can you repeat the question?
How can you prevent that other contaminants from the air affect the viability of the microorganisms that you use for the filter?
Well, if I can answer, the design of our biofilter, it is thought to have like a pre-treatment of the air section where the air could be filtered to ensure that no extra contaminants will enter to the design of our filter. So it would be like a mechanical filter, so we can ensure the bacteria won't like die or something like that, and the system will work as we wish it will work.
Okay,
Thank you. Great. So do we have any more questions from the judges?
Yes. Sorry. Maybe.
Marcela, please.
Sorry, I was mute. First of all, I want to congratulate you for this project. I think it's amazing. One of the things I like the most is how you took this opportunity to learn from experts in each phase of the design. So I want to know from this experience what has been the most valuable lesson for you.
Sorry, the most valuable lesson that the experts gave us related to the project, or
related to the experience to interact with experts in each phase of the design.
Okay, all right. Yes, so during this year, we talked with a lot of different people. We wanted to have two different approaches, people that are related with synthetic biology topics and experts in other areas. And when we talk with these people, we realize that the necessity to have a big approach of opinions, not just focus on science, but also talk about other things, especially the social aspects, because we know that we can have a very good project with a great synthetic biology circuit, but if it doesn't work in the real world, it is not going to be able to work in the market nor with society. So that's why we also wanted to talk with people that are experts in entrepreneurship and in the how to sell the project, and also in educational areas, and also about how to talk with people, how to talk with the affected communities by this problematic. So that's what I think that's the most important thing we learned, that we can't just focus on the science or synthetic biology. We need to have a wide approach and different types of opinions.
Okay, thank you.
Cynthia. Tanya, please.
Okay. So I agree with the judge. I really like your project. I think it's really nice and it's a good idea. So my question will be, what was the biggest hurdle or problem that you accomplished in your project, and why? So what's the problem that you had to come over to accomplish your project, and why?
Well, one of the first problem we faced was precisely like how to, because the air pollution is like a really wide problem. So how we could like focus on just the part we could solve, like reducing all the options we have, just to focus on that thing that we can manage. That was a really important part of our project, and also one of the most difficult things. Well, for me, it was one of the most difficult things to handle, but I think it was all worth it. I don't know if any of my teammates wanna add something.
I would like to, go ahead.
I would like to add that another difficult thing we had is the selection for the enzyme of the carbon genetic circuit, because it has the expression of one needs any methods that inhibit the expression of the other one. So it was very hard to find the correct enzymes for the circuit to work correctly.
Thank you. Let's go with the question from Sana.
Yeah, so hi. I really love the project idea. I just wanted to know how human practices formulated your direction. Like first, what were you thinking, and after, it's like solving local problems. How did human practices help to navigate your solutions into your project?
Okay, so during this year, the first thing that we did for our human practice approach was to make a survey, because we knew that, as Baruch said, the air pollution is a very big problem in the city, but we wanted to know in which points exactly of the city the problem is bigger. And with this survey, we realized that the places where the industries are, like the metal industry and all those industries, are the worst places where there is the air pollution. And we got to talk with these people, and they told us the health problems that they had, like allergies, skin problems, lung problems, everything. And that was the beginning. And then throughout the year, we got to start with the idea, the biofilter, and to get validation from experts. We got validation with an air quality management expert, with an expert in degradation of organic pollutants. But we also wanted to talk with people about entrepreneurship. So we also talked with a very important person from an enterprise that is called Green Friedrichs, that also makes biofilters, but for other things. And they also gave us feedback to how to make the project better in the commercial world. And we also talk with science educational experts, because we know that we can't just do a science project. We have to be able to communicate it correctly to the world. So he gave us feedback to communicate efficiently to adults and kids. So, and the math model. We also did a math model to be able to make the project better, to improve it, because we were not able to make experiments, so we had to make a math model to improve the project. So yes, I think that those were the most important parts of our human practices approach.
Okay, thank you.
Thank you. Marcela, please.
I think Angela just can do it first because this is my second question. So.
Okay, thank
Great.
you, Marcela. Yes, regarding the last question you were answering, did you have the opportunity to talk to, for example, local stakeholders in order to let them know the initiative? Because sometimes great initiatives are not taken to the practice just because will. So could you have some support for enterprises, for example, for trying the device?
Okay, so there are not a lot of biofilter enterprises in our city, but most projects are like startups. So that's why we contacted this. It's a startup that it's Green Fluidics, and we got a lot of how do you say apoyo? Yes, like the support. Sorry, like they support our project and they gave us a lot of feedback. But apart from that, there are not a lot of enterprises of biofilters here. So that really was a problem because we tried to contact like enterprises from other countries, but they don't always answer back. So that's why we changed our approach to contact more startups like this one. And according to but another important part from the local area is that we also wanted to talk to the affected communities. I think those two parts are the people that we contacted the most, the affected communities and the startups from Mexico.
Yes, thank you. We'll go to Marcela just to remind you, we have 10 minutes left.
Okay, I just want to know from the point of view of the team, one of the current trends in synthetic biology is the safety of using genetic modified microorganisms to solve environmental problems. So I would like to hear what is your opinion about that, if you consider that is a really safe strategy, what you think about this, because it's one of the trending topics around synthetic biology right now. So it will be to interest to hear that about you.
Yeah, so as you mentioned, yeah, the safety part of our project was really important for us. So with the validation of stakeholders, we could improve our biofilter in order to make a safe place and so the GMO can't sleep. Can escape to the biofilter, and also we contacted with stakeholders that could help us to make synthetic biology circuits on the safety area. For example, our kill switch will prevent our GMO in order not to escape, because if it escapes, then the GMO will die. So that's our approach to make our project to have safety implementations, and yeah, that's how we can say.
Besides, something really important to add is that the circuit, the enzymes we are using in the circuit, the original organisms we are taking it from, they are no pathogens. So, and we are putting everything into E. coli. That we know that E. coli is one of the most used bacteria in synthetic biology. So all of these, together with the kill switch my partner mentioned, it ensures that the project will be really safe.
Thank you. Let's hear from Sana, please.
Yeah, so following the question is like, what are the ethics in your country if you're introducing any GMOs? Have you taken that into consideration? Because the government and the policies are, it should be sanctioned by the government and the high authorities. So have you just built, or you just thought that we should approach someone from the government officials, and what are their concerns?
Does anyone from the
Uh,
team?
Okay, should I repeat? It's like how, like, what are the ethics regarding to GMOs in your country? Is it allowed? Is it not allowed? And what it is, because this is the GMOs you are just using, and synthetic biology. So yeah, just any information what your government or what your country thinks about it.
And we took into consideration the law of biosafety for genetically modified organism by Mexico. Este also some of the Semarnat that is in charge of for looking for the like the security of the biodiversity and stuff. We took the Cartagena Protocol, the Convention of Biological Diversity, that is like the first multilateral agreement for the biodiversity matter. Also the Nagoya Kuala Lumpur protocols. Yeah.
Okay. So you need to approach the government people for just sanctioning this. If it's there, if I like this project and I want this project to reach heights. So like that is the one thing that you will be thinking on it.
I didn't understand the question. Can you repeat it, please?
As in, you will be considering the GMOs guidelines as per your country.
Well, in our country, the synthetic biology part, it is not like too well explained. So we had to like take things like from the local law and the international law to make the consideration for our projects. We try to make it like the most well, yes, we try to take these things in consideration, but locally we don't have like an exact law that talks about the use of GMOs, so it was kind of complicated. But well, we are trying like to take into account all the antecedents we had to ensure we are like uh, uh, we are taking into account all the things we needed to take into account.
Yeah. Okay.
If I can complement the answer from Brooke.
Yes. Yes, just only to remind you, you have 4 minutes. So if any of the judges want to ask another question, please raise your hand to go after this answer. So go ahead, please.
Okay, sure. As well as considering local and international laws, the plan is also to biocontain the GMO in order to it do not escape the hardware. And if, as my partners already mentioned, if it does escape, it will automatically cease, it will die. So what we're trying to accomplish with this is like to prevent any legal implications regarding the how do I say it, the spreading of the GMO.
Okay. Yeah. So thank you all. Yeah. Next judge.
Thank you. Angela, please.
Yeah, thank you. Have you been able to evaluate efficiency of your devices? For example, how many would you need for a given amount of air cleaning, like CO transforming, in a given time?
No, as far as we could get in our math modeling, it was just how the behavior of our genetic circuit would be, but we don't have like an initial we couldn't get an initial like results on how we'll get how much will our system will degrade the contaminant compounds. So one of our objectives for future improvement of our project is to evaluate the enzymatic the catalytic activity of our enzymes in order to get better results to our project. So how will how will our biofilter works?
Okay, thank you.
Angela, do you have any follow-up questions for this? No.
No,
Okay,
no.
so we're just 1 minute left. So if there's no more questions, if there's anything from the team that you would like to share with the judges, this is your last minute, and then we will finish the session.
No, just thank you for your time. Thanks for all your questions, and well, thank you.
I would like about to say something about the question of the safety. Yeah, there's no like regulations like that in Mexico, but the Semarnat is the one that overlooks for the like the risk to human stuff. We got training about the official norms by Semarnat, the 087 for the microbiological waste, and for the 052 for the chemical waste, and about the toxins for the system. There's also no risk. Because it has been used implemented several times as a biocontention method in various projects because of being a bacterial elimination mechanism originated in E. coli. So it should not represent also any risk to humans. So yeah.
Yes, thank you. Well, thank you, everyone. Thank you to everyone from the team. We're very excited to have you here and for you to be participating. So your session is over. We're going to ask you to please leave the room to give access to the next team. Thank you, and thank you for your questions for the judges. We will have a 5-minute break to bring the next team.
Thank you.
Thank you very much.
Thank you.
Thank you.
Okay, so for now, this was our first session. So now we are waiting for the new team to start joining. We have a couple of minutes. So if you need a break, this is the time to do it. And we will be accepting them in 2 minutes.
Yes, thank you.
Hi,
Thank you.
we are
Oh,
now letting the BioVerse team in. Let us know if there are any other team members that need to be accepted.
okay, so while we wait for the whole team to be here, I just want to welcome you all. Thank you for participating, the judges and the team in this judging session. We will start in 1 minute. So if the team members can please open your cameras. I will be sharing your promotional video in 1 minute, and after that you will have 20 minutes to answer questions from the judges. So please be mindful with your time and with the judges' time. Thank you. We will start with the video from the BioVerse team.
Tienen semanas sin ver agua saliendo del grifo. Deseamos que llueva. El río Jamapa no ha subido más que 20 centímetros.
De la noche a la mañana, Monterrey descubrió que el agua no es infinita.
The world faces a water crisis. This year, Mexico has suffered one of the most extreme droughts. In central Mexico lies the Pico de Orizaba, the tallest mountain in the country. The Pico de Orizaba is a natural water tower providing water relief downstream for more than 2 million people. However, it has lost most of its glaciers that slowly release water during drought. Forests that once retained water have been replaced with eroded soil due to deforestation. Water scarcity starts here, but so does the solution.
The BioVerse, an interdisciplinary team of students, aims to restore the water cycle and ecosystem with synthetic biology by tackling 3 problems: drought, thaw, and water retention. We focus on a dual production system. Not only can our engineered bacteria efficiently generate 1 world-changing product, but 2. The bacteria will express a membrane protein with the best ice-nucleating properties, freezing water at higher temperatures, maintaining and regenerating glaciers. Not only that, but ice nucleation can be potentially used for cloud seeding. Secondly, they will produce an eco-friendly exopolysaccharide, capable of retaining water and stabilizing soil, promoting vegetation growth.
This way, we can achieve an efficient use of resources that are usually thrown out, such as bacterial cells. And being fed with agricultural waste, products are significantly reduced in cost. Our product is not limited to mountainous regions. Stakeholders like farmers can also benefit from our product, helping crops thrive in drought.
Thank you to everyone from the team. So we will start with questions from the judges. Please raise your hand if you want to ask a question. Yes, we will start with Juan Pablo, please.
Okay, hi guys. First, I would like to say that the idea seems to me very different from is normally done or proposed for this type of projects, to be honest. For that reason, I would like to know what made you came up with this proposal?
Anyone from the team who would like to answer the question?
Yes, so we are a very wide range of students, and we're from different, we're all the way from high school to college. So not all of us had that expert knowledge or more deep knowledge on biology. So we took that as an advantage because when people are more knowledgeable about something, they tend to be biased to what is already there and to the systems that they know. But we saw that more people that didn't have like knowledge, strict knowledge on biology, were more likely to have like that creativity and to not limit themselves. So we created this type of like crowdsourcing with everyone, and we started building off that idea of thaw and water retention, and then deepening it and like collaborating like with the part of biology to deepen that idea and make it like creative, but also like information based.
Okay, thanks.
Thank you. Let's go with Angela, please.
Hi, yes, I want to congratulate you as same as Juan Pablo. I think that is a super novel initiative and clearly represents a great research effort. But from my point of view, if that was Colombia, for example, it might represent a counter incentive. You know, deforestation is the greatest problem maybe there. And if people, the local or the people interacting with the ecosystem will see that there is something that will retain water in the soil, maybe deforestation stops being the problem. So nothing is going to be done to interact and solve that problem. Have you thought that it could be taken as a perverse incentive to follow deforestation in the area?
Yes, there's someone from the team would like to answer the question.
Yeah, sure. I think it's a really interesting question because we had that thought, especially since we wanted to make it worldwide and accessible to everybody. But for now, we have made it especially for Mexico, which is the case for now. But if we continue, and I'm sure everybody in the team would love to continue with our initiative, we would love to be able to make it so it's not a problem, because maybe for other places it could be perceived as different, but for in the case of Mexico, we're taking it as an advantage. And maybe, who knows, we can make it for everyone, accessible for every single region of the world. And that's our goal.
Thank you very much.
Thank you. Let's go with the question from Tanya.
Okay. Hi, team. I really like your project. I think it was really cool idea to prevent the global warming. But I have a question about if you would have the chance to come back again to your project, which things will you be doing it differently?
I'm sorry, could you repeat the question, please?
Like if you, because now you have the experience and you went through the whole process of developing the project. So if you have to go back and if you have the chance to go back, which things will you do different?
I think one of the main things that we would consider is the information that we research about the build and test, like tests for making our project more viable to do at an experimental level and therefore at a production level. I think that we would research more about that and find ways to be more, for our project to be more viable, in a sense of finding different biobricks or different biological parts that can help us to be more, for our circuit to be more efficient and therefore to be able to be constructed in a larger scale so that we can really make an impact in our customers or the people that we're trying to help around the Pico de Orizaba.
Perfect, thank you.
Thank you very much. Let's go with the question from Sana, please.
Yeah, so hi, it's just a very novel idea and really I like the project. So as you mentioned about the entrepreneurship model, so if you got a patent or you got an opportunity to build up the entrepreneurship model, would you like to go for it, and will you interact with stakeholders, and what are you planning on the business model of this project?
Yes, we would like to elaborate more in that area of entrepreneurship. We had a late, we're going to have like a late interaction with industries that are related to sugarcane. So we can take that into the process of the circular economy that was mentioned in the juggle. So we wanted to elaborate more on that because our project, it was more focused, it has a great potential for more like industrial purposes, as was mentioned for like the production of a different exopolysaccharide or other biopolymers that are currently produced in low quantities and therefore are very expensive. So yes, we want to expand this area, and we would like, we're going to have these other meetings to open our panorama to the industry.
Okay, all the best and thank you.
Thank you. Marcela, please.
Hello. First of all, I want to congratulate you because I think it's an amazing project to be developed by high school and college students. And I think it's this is the spirit of IGN program. So I think it's amazing. I have a question because it's not clear for me how you imagine you are going to apply your synthetic construct on the environment to solve the problem of the Pico Orizaba. Are you expecting to release the bacteria, or are you expecting to release the biopolymer? I want to understand better what is the idea that you are imagining with this.
Yeah, so our bioproduction is based, well, on the dual production. So the biopolymer, the xanthan gum, will be separated from the actual bacteria. So that is like one of our products. So it's going to be the biopolymer in a in like a powdered form. This can be applied for agricultural purposes and the deforested areas of the Pico de Orizaba. On the other hand, we have the product that's going to be like the ghosted bacteria with the INAS on their membranes. So this will also come in a powdered presentation, but it's not releasing the bacteria alive. It's just going to be like the membrane with the INAS proteins.
Do you have any follow-up question, Marcelo?
No, that's okay, thank you.
Okay, let's go with the question from Juan Pablo then.
Hey, I would like to know, why did you choose to work with xanthan gum? I mean, there is another biopolymers that can be used to retain water. So I would like to know what factors were taken into account to work with this biopolymer.
Yes, before we go to the team's answer, I just want to let you know we have 10 minutes left. So just be mindful with your time. Thank you.
Okay, so we chose xanthan gum because we noticed, well, through reading through going through the literature, it was mentioned that xanthan gum does not only have water retention properties, but it also has properties that decrease or increase the health of the soil by putting together the soil particles. So this decreases the erosion from water and from the air. So this is another like benefit of xanthan gum, but it also increases the vegetation index. And there are many reports on xanthan gum increasing vegetation index. So we also found it applicable for areas of deforestation where there is a lot of erosion, no vegetation, and where water needed to be retained. So it has more benefits than other polymers that we investigated.
And do you take into account the economic factor? I mean, the cost of the xanthan production?
Yes. Well, actually, we thought that it was a good opportunity to show like a solution, because right now the xanthan production, the xanthan gum production, it's very costly, and it's especially in certain industries because it's not available in a lot of countries. So there's like little industries that take up all the market, and therefore it's expensive. But on our research, we found that there were a lot of different points that could be more efficient. And by proposing a biological circuit that can produce xanthan gum without the need of fermentation, for example, that it's one of the main like processes that it's used right now to produce xanthan gum, we thought that it was a good opportunity to actually go into the market and propose a solution that can decrease the cost of well, the action production at the moment.
Okay, thank you.
Great, thank you. Are there any other questions from the judges? Yes, Anna.
Yeah, so you mentioned about like PFK, phosphofructokinase. You are using an alternate pathway. So what's the relation between glycolysis and PFK and the project that you're using, that the normal glycolysis, and what alterations you are doing with or alterating the PFK module in your project, as you mentioned?
Yes, so the xanthomona metabolism, it has like three main routes to go to like the pyruvate, which is the ED pathway, the pentose phosphate pathway, and the normal like glycolysis. And for this, we found that xanthomona didn't have that much flux through that pathway. And there's been some reports that state that this is because, or mostly because, PFK in xanthomona is a conserved gene and it isn't expressed as much as in other bacteria. So they added, in these reports, they added PFK to xanthomona, and there was an increased growth rate. So this is what we wanted to do to reduce the time of growth. So we could get to the stationary phase faster, so production can be faster. We introduced the PFK from E. coli K12, which is PFKB. Although it is expressed at a lower level, usually, it is expressed at a higher level when PFKA, which is the PFK that xanthomona has. When this PFKA is expressed at a lower level, then PFKB is expressed more. So this was a good balance for us to increase flux through glycolysis and therefore increase growth rate.
Okay, and you mentioned about the inhibitors of ED pathway that are being used. So what inhibitors are you like, is it synthetic inhibitors or it's any natural inhibitors of ED pathway?
I'm sorry, could you repeat the question, please?
As in you said about the biochemistry. So it was just mentioned like the inhibitors of the ED pathway being used. So what inhibitors of the ED pathway are being used in your project?
Okay, so we use the ED pathway to, in comparison with other biopolymer production, the ED pathway contains various forms of sugars that can also be used by just adding an enzyme to our sequence. And these like products of sugar can be used to create these biopolymers. So we didn't block automatically like the ED pathway because it is important also for detoxification. Plus, we can use the ED pathway to produce sugars that can be used to create other biopolymers.
Okay, thank you.
Thank you, everyone. Just so we have now less than 5 minutes left. So if the judges have more questions, please do your final question now so the team can answer. Yes, Tanya.
So my question it will be more about how are you planning to continue? Are you planning to make your constructs a reality?
Well, yes, we're trying to first actually trying to make like a plan for us to work and to, for example, as it was said before, like an something more related to entrepreneurship, so that we can actually form a plan and there and then start working on how we can make the project more viable in a lab or doing the experiments necessary so that it can be more attractive to be able to introduce it to a market that it's viable and that it can be more well, that it can impact people directly in our community. So first we're going to base our we're going to base our project in an entrepreneurship perspective so that we can actually know what are the plan and how we can manage to get the enough resources so that we can start actually designing how we're going to work in a lab to make it possible.
Yeah, I suggest you to begin working like maybe in parallel, because something is to do some design in silico, but when you have to go to the lab, then you need to make it, you really need to make it happen.
Yeah.
So it's like,
well, yeah, because we also consider this part because we have to analyze different reagents or if the parts that we chose are going to be efficient at an experimental level. So I think that you're right. It would be important to make them at the same time. I mean, I think firstly the experimental part, but yes.
Thank you. Any more questions from the judges? We have 2 minutes, so we have time for one last question. Okay, so if we don't have any more questions, we just want to thank the team for coming in today, and we will let you go. Thank you. We're excited for you to be participating in this competition, and that's, well, just wait for the results in the following days. Thank you very much. So please just leave the room. We will make the space to admit the next team. Thank you.
Thank you so much.
Thank you so much. It was a pleasure.
So we have, again, we have a 5-minute break to wait for the next team to show up. Thank you. Hey everyone, so we are waiting for the next team to join. In 1 minute, I will start sharing the video for our next team, that's IGEM and NAWAC. And then the team will have 20 minutes to answer the questions.
Hi, Jocelyn. Could you ask your teammates to rename themselves so we can identify if they're from your team on the Zoom waiting room?
Hey, Jocelyn, can you share with us the name of your team members so we can give them access?
Can you repeat the question, please? I don't listen very well because my internet is
Yes, thank you. Can you ask your team members to please put the name of your team in their names so we can give them access?
yes, they have the change the name.
Okay. Well,
in
thank you.
1 minute they connect.
Okay, great, thank you. So we're waiting for the team. We'll wait for 1 minute and then we'll start with your video. So the judges can see the video while you join the room. Okay, I will start sharing the video now.
Water is a fundamental part of life as we know it. Every organism needs water to some extent. The human body is approximately 70% water. This water is constantly being recycled all throughout the body. As one of our human rights, we all should have access to safe potable water. Society's accelerated production and consumption of pharmaceuticals means that they are also going to be disposed of in the environment. As a result, aquifers are continuously exposed to a significant load of these compounds that exceed domestic and industrial purification capacity.
The Mexico City metropolitan area is one of the most affected by pollution and was declared the most polluted city in the world in 1991. Por San Martín is an important water source in Estado de Mexico, supplying around 155,000 people. Recent studies have demonstrated that water in this dam is highly contaminated. The presence of emerging pollutants such as various drugs has been determined in wastewater and sludge from Mexico City metropolitan area.
Our team has proposed to combat this contamination by developing a genetically modified organism, using as expression vector Pichia pastoris and genes from white rot fungi such as Trametes versicolor, due to its specificity in the exterior to attack substrates through the action of intracellular enzymes from cytochrome P450 system and extracellular enzymes such as lacases and peroxidases. This is what makes these microorganisms efficient degraders of xenobiotics.
The capacity of degradation of persistent organic pollutants in water by different varieties of fungi has been previously studied in several investigations. We strongly believe that everyone should have access to clean water, free of any pathogens and contaminants. That is why, with our project, Both the local inhabitants and those communities that receive water services from the Marin Dam will be able to obtain clean water free of the main polluting pharmaceutical compounds responsible for various health issues.
Great, thank you. So now that we have seen your video, thank you to all the team members that are here. You have 20 minutes to talk with the judges. They will be asking questions, so please raise your hands if you have any question. And for the team members, please be mindful with your time because we have a very short amount of time. So if any judge want to ask a question, please. Yes, Sana, please.
So hi everyone. I really like the project and all the best wishes. I just wanted to know why you choose Pichia pastoris as in, is the model organism or anything, the advantages of using Pichia in your project.
Thank you. Does anyone from the team want to answer the question? Yes, Christopher, go ahead.
First off, thank you for your question. We chose Pichia because of its versatility. It's a well-studied platform. So our advisors, we had a different option as well. Our advisors suggested that we use Pichia.
Okay. Thank you.
You're welcome. Thank you.
Thank you, Marcela.
Hello, everyone. First of all, congratulations for embrace this opportunity. My question is very simple. Lacazar enzymes that own a wide range of compounds, and they are actually used in many strategies of bioremediation of a different kind of compounds of pollutants. So, do you think this could be advantage or a barrier to develop your project that is not really unspecific targets? Lacases and peroxidasas can act on a wide range of compounds. So do you think this is an advantage of your design, or it could be a barrier of that?
Yes, Sandra, go ahead.
We think it can be an advantage because it is not so specific, so it can also help degrade another pollutants present in the dam.
Okay.
Thank you. Do you have any follow-up question, Marcela?
Yes, I would like to know what was the team experience on the interact with the community, socializing their idea of the project.
Is there anyone from the team who would like to answer the question? Yes, sure, go, Fabiola.
So for choosing our project, we talked to people that have worked near Presa Madin or that live near Presa Madin. And that's how we chose and narrowed down our spot. And also because it's kind of close to our university. But we plan on continuing this project and to keep having interaction with the community, visiting them to help them in other ways that don't involve our project, such as by something called video aquatico, I don't know how to say. So by cleaning it with our own hands and everything. So we talk to people that live there, have worked there, and we plan on doing it.
Okay, thank you.
Thank you, Juan Pablo.
Hey guys, first I would like to know what treatments have been previously implemented at the dam that haven't not worked right now. And secondly, I would like to know a little more about how do you plan to implement your proposal in the dam?
I can answer the first one. So a treatment per se has not been implemented. They try to, well, they're building a water treatment plant, but they have been building it for a long time and it's not working. We don't know the actual reason. No one tells it like punctually. So we don't know why it's not working and when do they plan to like open it. But we don't think that's going to happen anytime soon. So the things they have done to clean it are things that I have mentioned before. So like cleaning it with volunteers that want to help take out the plants that are invading it and things like that, but nothing like our system has been tried.
Thank you. Yes, Edwin, go ahead.
May I add? Okay. Well, another important aspect of this is that the pharmaceutical compounds are not regulated as any other pollutants in the treatment plants, such as the heavy metals or or coliforms that are present, and they are much more focused in bioremediating this. And the implementation, we believe that our organism will can be in a matrix in a part of the treatment plant, and and then the organism will do the cleaning.
Juan Pablo, I don't know if you have a follow-up question.
Yes, the last one, it will be how you are going to implement the proposal. I mean, are you going to produce the enzymes and release it in the dam, or are you going to build a filter with PICA?
We're aiming to make an immobilization device with the PICA pastoris and implement this in a part of the treatment plant.
Okay,
Thank you.
thank you.
Okay, let's go with the question from Tanya.
I really like the project. It was interesting. But my question is more related about the human practices. How did your human practices activities evolve with your project?
Does anyone from the team who would like to answer the question? Yes, Edwin.
Yeah, as Fabiola mentioned it, to decide our problematic, we did research and some talking with members of the community. But I think our human practices this year was more focused in all the creation of the team with it was inside the university to let them know about us, about the team, and what we made is to help them raising money for the fee, etc. So it was like the first step to getting the university to know the iGEM. So that's what we were more focused in the human practice part.
Tanya, do you have any follow-up question?
Yeah, so my following question will be how, if you have plans for making the experiments happen, or how do you see the project is going, how do you envision your project to continue?
Yeah, sure, Sandra, go ahead.
Well, we would first have to synthesize the plasmid, then propagate it, then transform our Picha pastori cells. And before the implementation in the dam, we could experiment with our transformed cells in a bioreactor that mimics the conditions of the dam to see how it works. And if it is favorable after all the statistical analysis, we could implement them in the dam.
So you are planning to continue doing experiments. So your team will not disappear. So you're planning to continue working together.
Well, we would have to have the support of our school to do so, but it would be ideal.
Great, thank you. So just to remind you, we have 10 minutes left. So if anyone else from the judges have any other question, please just raise your hand. Yes, Anna.
Yeah, so you mentioned like hydrolysis and peroxidases in your promo video. It will solve the problem. So any comments on that? Why you're choosing that the biochemistry pathway of hydrolysis and peroxidases, and how it will affect your project?
Does anyone from the team who would like to take this question?
I'll take it. Peroxidases are also contemplated as being advantageous for us in our project. Since our project is based around bioremediation and the degradation of pollutants, such as pharmaceuticals in this case, there are different mechanisms through which you can achieve that goal. And since peroxidases do have a different mechanism of action, I think it would be a good option for us to explore in the future to incorporate into our platform. And this would, in the end, give us a broader field of action, and in the end would be beneficial for our goal, since we would achieve cleaner water.
Okay, and one more question, how it will affect the bioremediation process, as in your project, in one line, as in what are the benefits if any entrepreneur stakeholder want to accept your project or sanction it within the local communities or the government or the SDGs? So what is one of benefit that you will say, okay, this is the advantage of our project? Is
Okay,
it, yeah.
good question. Thank you. I think, especially in Mexico, with the current government, there has been, I wouldn't like to call it a problem, but a suppression of the scientific community. So it would also serve an academic purpose in our society and in our social context. So as long as we stay involved with the community, I think being exposed to complex subjects such as bioremediation through microorganisms, I think we would be working to counteract all the damage that has been done to academia and education. So I think a progressive government, maybe locally, could be interested in our project, and that would be a step in the right direction, I think, to be a model of how progressive science can benefit our society and culture in general, to, in the end, remediate past past errors, such as contaminating our water sources.
Okay, thank you.
You're very
Thank
welcome.
you. Marcela, did you have another question?
No, it was very similar to the one that Sana did. So
Okay, no problem. So we have 5 minutes left. So if the judges want to ask for one last question, for a few last questions for the teams to answer, please go ahead. Yes, Angela.
Hi, thank you for your answers. Despite the real possibility of implementing your project and all the context you gave us about the government and practical issues, how would you tackle with the outreach of your project? Suppose it is gonna work, it is gonna be implemented. How are you going to communicate it to people in order to reach your objective, which was clean water for everyone around the dam?
Thank you for your question. Also, a very good one. I think, unfortunately, one of our human practices members couldn't be here, but I think through human practices we could achieve that, especially through social media. I think it's complicated because expressing technical scientific knowledge is complicated to people that aren't exposed to it regularly. So I think we'd have to find a way to bridge our scientific knowledge with popular understanding. So I think there's a great opportunity to bring other people with different abilities and knowledges, to bring them on board to our project, because in the end, as you say, doing the project isn't just doing the project. People have to know about it as well.
May I add or something pretty quick?
Yes, please.
Sure, go ahead.
Well, also, as Fabiola mentioned earlier, our university makes these like annual cleans of the dam, which they go in kayaks and they clean it with their hands, and so we can join with them because it is made by other careers. So this is one of the ways we can engage with the community, by going to these activities that are made in Madin Dam, so we can participate in order to let them know that we are not only cleaning it physically, but we're only also aiming to clean it in like scientific approaches. So that will be one of the ways that our university, since it is close to our university, the dam, there are many activities being held in there throughout the year.
Also, sorry, also before this project started, well, I'm not from biotech, I'm from engineering. So there were a lot of foreign concepts that I really didn't understand. And I was able to understand them because, well, the team that really did know them were able to put them like to a child's level. So someone that isn't really familiar with it can understand it. And I think we could replicate this model as to how they explained things to me, to them, because they are also people that really don't have this background and don't really understand, and probably have a worse background than me because I have had some kind of contact with science. So by doing this, we can reach, well, these people and be able to share all the things we were able to learn, and to tell them, yes, this is really going to work because this is what we're going to do. And I think if we take them to this level, we will be able to share our knowledge and really help the inhabitants of Medellin Dam.
Okay, thank you.
Thank you, everyone. So we're just running out of time. So all that's left is to thank everyone from the team who were able to join us. Thank you for your time and also for the judges for your mindful questions. So we will ask the team members to please leave the Zoom room and just continue following up in the YouTube live stream for the next team. We will have again a 5-minute break waiting for the next team to join in. Thank you.
Thank you.
Bye.
Thank you, everyone.
Thank you.
Have a nice day.
Bye.
Hi, Ana Laura. Please remind your team to change their name so we know that they're from your team and we can let them in. Thank you.
Okay, thank you. I will remind them.
Welcome everyone from the ICANN Connect team. I hope I'm pronouncing it well. We will be starting your session in 2 minutes, so please, everyone from the team, just rename yourselves so you can have the name of the team also in your name. What we will do is that we will share your promotional video for the first 2 minutes, and then you will have 20 minutes to answer the questions from the judges. We'll start in 1 minute. Thank you.
Okay, thank you.
Thank you to the team members. Angela had to leave, but she's asking for you to ask one question that she left. So if anyone from the judges know which is this question, just let us know. Thank you, Marcela. Great. Okay, so I will start sharing the video in 1 minute.
Hello, Grandpa.
Hello, Gaspar. How are you?
I'm sad because I read in the news about a lake that used to be very beautiful and magical, but now it's just contaminated and ugly.
Let me tell you a little story about that lake. When I was young, we used to go down on the lake to a lot of my family and friends. Tourists from all over the world tried to see it. The textile and fishing industry were the major source of income for a lot of families around the lake. But eventually, as the town grew, so did the problems. The lake became a deposit for waste and residual water. This water contains nutrients that enhance the growth of harmful microorganisms. And that's the story about that lake.
In the end of this lesson, we can conclude that bioluminescence is an amazing and important feature in some animals.
What happened to you? You look like you just submerged in something green and ugly.
I used swimming in the lake. Bad idea.
You know, a few years ago, my grandpa talked about the lake and it sounded so different from what we see now.
Do you think you have microorganisms on you?
That's for sure.
Do you think we can use them and take advantage of them to use them for something useful?
I actually don't know.
Well, nothing is impossible if you think science.
So the plan is to make them go, and maybe you could also do something so they could clean the lake.
And they could also produce pigments useful in the textile industry.
Yes, I think we got it.
If you believe in synthetic biology, join us in what could be a game changer for an efficient use of microorganisms of contaminated lakes all over the world.
Great, thank you. So now we will have 20 minutes to answer the questions from the judges. So if any judge wants to start, please raise your hand so you can ask your question. Yes, Juan Pablo, please.
Okay, hi guys. Thank you for your presentation. I really enjoyed reading the proposal. I understand that from the project that the cyanobacteria are already found at the lake. And yet there are problems of eutrophication. Indeed, I believe that its presence is due precisely to eutrophication. If so, why would transform it reduce the problem? I mean, considering that the modification is more from an aesthetic point of view in order to increase the tourists in the lake. So while transform the cyanobacteria, it would reduce the problem of eutrophication.
Yes, Luigi, go ahead.
Hi, thank you. Well, what we thought was to use the same cyanobacteria that already exist in the lake so that it can consume the nutrients and avoid the nutrients to be like available in the lake. So the cyanobacteria is not intended to be used in the lake, but in an isolated system, like in the bioreactor that we mentioned in the proposal, so the cyanobacteria can grow in the bioreactor and reduce the concentration of pollutants in the lake so that they are not available for the cyanobacteria bloom in the lake, so that it just grows in the isolated bioreactor while expresses the bioluminescence as a touristic attraction. And then we can collect the biomass to extract pigments and other uses, but not keeping the cyanobacteria in the lake. So we are given like better conditions. We are trying to use the cyanobacteria from the lake because it will be adapted to the lake conditions, but we will give them the enough air so that it grows and consumes the nutrients, reducing the loading of nutrients in the lake.
Yes, but the water that you are going to use in the bioreactor is the same water that is in the lake. And the cyanobacteria that you are going to use are the same. So what is the difference to use the cyanobacteria in the lake or in the bioreactor if the only difference is the transformation that you are proposing to get to produce light?
Yeah, it's just to improve their growth by giving them the air necessary and avoid the growth in the lake and allow the growth in the bioreactor with better conditions so that it can consume the nutrients, because they can do already in the lake, but we plan to use the bioreactor in the entrance in the outlet of a river so that the nutrients do not enter to the lake because the cyanobacteria are already in the lake.
Okay.
Thank you. Marcela, please.
Hi, everyone. Congratulations for this proposal. I think it's very robust. I'm going to do a lot of questions, one for me and the other one for another, just that you have to leave before. So first of all, I have concerns. What is the strategy to manage the biomass that you are producing in the bioreactor and it's not acting anymore? So what are you going to do with that apart from the extraction of the pigments? So because if you are going to extract the pigments, you are going to need to kill the biomass. So you are going to feed the bioreactor continuously. So what is the strategy to do that?
Yes, Luigi, go ahead.
Well, the plan is to use part of the biomass to extract pigment, but not all the biomass. We plan to use to have a recirculation to get part of the biomass for the initial inoculum that enters in the entrance of the bioreactor. And all the biomass all the biomass will be collected from the water by a centrifugation method to get part of the biomass for the pigment extraction and part for another inoculum for the entrance of the bioreactor. And all the water will be treated from a filter and then through a UV and ozonolysis method so that no cyanobacteria gets back to the water nor plasmids, and just the water without the biomass.
Okay. Okay, so I'm going to read a question that leave Angela. There is a judge that you have to leave. So she is saying, I am concerned about the final outcome, the bioreactor around the lake and the light. I understand from the video that it will be located outside the lake. So the light produced can be seen and enjoyed by tourists, but also can be seen by locals and local fauna. Is there native fauna remaining in the area around the lake that can be affected by that nocturnal light that you are going to produce?
Yes, is there anyone from the team who would like to answer the question? Yes, go ahead.
Yes, the the light is like for the tourist attraction. We did research and we didn't find any like species that would be affected. Basically, because the light, well, the shore of the lake is already illuminated, and all the species are going to be used to that lighting. So there would not be like a difference in if we put a light like in the middle, the light in the middle of the lake. But since it's gonna be in like in the shore, well, there already exists light there. The difference is that it's gonna be like natural light to attract tourism back to the lake.
Okay. And my last question is about the safety strategies. What is the strategy to the safety that you consider? Because you can have a filtering of the biomass from the bioreactor to the lake. So what are the safety strategies that are you thinking to go ahead with the project?
Yes, go ahead.
Yeah, the main strategy is that to make the centrifugation, to separate the biomass. Then if some bacteria still remains in the water, filter it, and then the water passes through an ozonolysis and UV lining, so to kill all the cyanobacteria and plasmid that can be still in the lake. We also propose to have like a double wall bioreactor, so that it has a leakage in one joint, or it breaks in some part. There's another layer to keep the water, and also to keep several valves along the bioreactor, so that if there's a problem in one part, in one section of the bioreactor, we can close it and go to fix it instead of letting all the water get out of the system. And we also plan like to talk to the community and tell them the benefits and the risk of the cyanobacteria, so that they can help us also with if they notice like a problem in the system, so that they can report it. Besides the monitoring that the system has to have, and like in the system, those are the things we have thought about, and beyond all the laboratory protocols and those things.
Okay, thank you.
Thank you. So we have 10 minutes left, and we'll go with the questions from the next judges. So Juan Pablo, please go ahead, and then we'll have Sana and Tanya.
Okay, I would like to know if you have established the cost of use UV light and ozonolysis as a methodology to kill the bacteria and avoid to get the lake. And I ask that because I don't know if you have do some research about other methods to remove these nutrients in order to compare if it will be economical viable, the project.
Yes, anyone from the team can answer the question. Yeah, yes, just go ahead. No.
Celeste or me?
Yeah, yes, if
If you
you're
want,
from the team, you can answer.
Well, actually, we did not get like to the cost measuring. We like designed the sizes of the equipment, but we did not like, we know like the methodologies to get the costs, but we did not have time to do that for the project. So we actually do not know the exact cost for the size of the system. And yeah, we also did research about other methods. For example, in Guatemala, they're planning to use like the a nanobubble system, but it really does not eliminates all the concentration of the pollutants, and it's still not working. So that's why we thought about this new project to treat it. But yeah, it's still necessary to make all the economic analysis with the investment and the production and the operating costs versus the benefit. That is mainly a bioremediation project and more than a profitable project, but we also plan to take advantage from the pigment extraction process and support from the community and municipalities.
Okay, thank you.
Great, Sana, please.
Yeah, hello all. I really like the project proposal on Joggle. So I would more glad to know, like, is there any field studies are you planning, or is there any prototype of field studies, because eutrophication or bioremediation, it has an environmental application. So what are your take on this?
Yes, anyone from the team can answer. Go ahead, please.
Yes, we plan like to make profitable this project, making these pigment extractions and selling them to the local industrial. We are planning to make like dye workshops and these things, so the community can make profitable this plan, this project that we will set in.
Thank you.
Okay, and
no, go ahead, Sana.
okay. So yeah, it's just a follow-up question. Like, is there any problem in the extraction of pigments? Are all the pigments soluble or insoluble? Because if it's insoluble, then it will be a lot of process involved. So the pigment that you have elaborated on Joggle, all the pigments are soluble or it's insoluble, and how you're tackling the pigments in the environment.
Yes, is there anyone from the team? Yes, go ahead, Luigi.
Well, we like plan to analyze what are the specific pigments of the project. We know that this cyanobacteria can produce fish picocyanin, which is like it's a protein pigment. So we plan to extract it from different methods, including precipitation and also separation in chromatography. Also, it's got chlorophyll and other carotenoids. So first, we like plan to use a method to know what specific pigment the isolated cyanobacteria can produce, and then analyze like the specific process to recover all the pigments by being whether soluble or insoluble pigments, depending on the pigments we can extract, considering the native cyanobacteria that, well, we haven't isolated, and the changes that could induce the modification. So to know which pigments are mostly expressed, and then scale up the system for the extraction. But since we do not have the specific cyanobacteria from the lake and what changes, it can suffer from modification. We don't know exactly the pigments. We, like in the proposal, plan to do the scaling for C-cosinin and chlorophyll, but yeah.
Okay.
Thank
Yeah. Thank you. Yeah.
Thank
Sorry, Tanya.
Yes, go ahead, Tane. We have 3.5 minutes left, so
Okay.
this
super.
probably will be the last question.
Okay, so my question is more about like what advice would you give to other teams who want to work on kind of a similar project like yours?
Yes, Ana Laura, please go ahead.
I think I would advise them to research as many things as possible because we don't know what things can affect other things. So I would say that they should really research deeply into all of the things that their project could benefit, but it could also be harmful so that the impact is less. So I think research is a very, very important part.
Do you have any follow-up question, Tanya? We have time for one last one.
It's okay. No, it's fine. Thank you.
Great. Okay, so if there are no more questions, I don't know if we have two remaining judges, Sana and Marcela. If you have any other question, please ask now.
No, not for me.
Okay, great, thank you. Okay. Oh, Sana, I think you're muted.
Yeah. I said no. It's okay. Thank you.
Okay, great, thank you. So we have only 1 minute left, so that will be it. Very thankful for the judges who had the time to be with us in this session. Thank you very much for all your questions and insights. Thank you to the team members for participating in the competition and in this judging session. It was great to see you here. And we will leave this Zoom room now, and we will have a half an hour break, and then we will continue with the next session.